Hey everyone, and welcome to RUBIN'S Mind Lab, where we break down the latest research into what it really means. Ever wonder how scientists can make sure their amazing discoveries about the brain and behavior are actually real, and not just a fluke? Well, today we're diving into a super cool R package called ggsem that is all about making sure scientific visuals are clear and trustworthy. Think about it. Scientists spend tons of time analyzing data and creating fancy charts and diagrams to show their findings, but sometimes comparing different versions of these diagrams, or making them look just right, can be a messy manual process. This paper introduces a new tool, ggsem, that aims to fix this. It is designed to help researchers create and compare these visual models in a way that is super reproducible. What that means is, if another scientist wants to check your work or build on it, they can easily recreate your exact visuals and even understand all the tiny choices you made along the way. So the big problem these researchers wanted to solve is this. When scientists compare different models of how things work, like the brain or people's behavior, they often end up with a bunch of separate pictures. Trying to make those pictures match up perfectly so you can really see the differences and similarities is a lot of work. You might have to tweak layouts, change colors, add labels, and do it all by hand or with lots of separate code. This can be a real headache when you are trying to make sure everything is accurate and can be repeated. They explain that even though there are great computer programs out there to help with the math behind these models, the part where you actually show the results visually is still a roadblock. The tools they have are great for showing just one model, but when you want to show lots of models side by side to compare them, it gets tricky. This means scientists often end up fiddling with their pictures outside of the main analysis. They might use a separate drawing program or write extra code only after they've looked at the first picture. The problem here is that these visual tweaks get separated from the actual numbers and statistical models. When you change the picture, you might lose the direct link to the statistics that created it. And if it is not linked, it is really hard to perfectly recreate it later or understand why you made certain visual choices. This is where their amazing new tool, ggsem, comes in. Instead of just messing with the drawing itself, ggsem focuses on the statistical parameters. Think of parameters as the numbers or relationships that describe how things are connected in a model. ggsem lets you directly tweak the visuals based on these parameters. And here is the really cool part: it saves everything you do. All the changes you make, like a detailed history book, this history is called metadata. This metadata is structured in a way that you can trace back and regenerate your figures perfectly. It also plays nice with other tools scientists already use. This figure here really sums it up. On the left, you see the old way, lots of separate plots. Then you try to stick them together. It is all disconnected. On the right, with ggsem, it is a unified workspace. You interact with the statistical parts and the tool keeps track of everything. They build ggsem with three key ideas in mind. First, parameter-linked interaction. This means you are directly working with the statistical parts of your model, not just the lines and boxes on a screen. Second, interoperability. ggsem can work with information from a bunch of different popular R packages that scientists use for statistics, so you don't have to switch tools completely. And third, metadata-based reproducibility. This is that amazing history, keeping feature. Every change you make, every decision is saved as structured data. This means you can undo, redo, save your whole session, and then bring it all back later or share it. So how does it actually work? You start by loading your statistical models into ggsem. These models can come from various R packages. If you are just working with one model, you can load it directly. But if you want to compare multiple models, which is often the case, they have this neat builder interface. It is like a step-by-step guide. You add each model you want to compare, one by one. You can even tell ggsem where to place each diagram on the screen, like next to each other or one above the other. This is great for making side-by-side comparisons. And remember that metadata I mentioned? Well, each diagram you add gets its own metadata. This includes all the details about how it looks and all the editing steps you took. When you start making changes in ggsem, like making a certain line thicker or a node a different color, it doesn't just change the picture. It records why you change it and associates that change with a specific statistical parameter. This is key to reproducibility. And the final magic trick is that you can take this saved metadata and turn it back into a high-quality image, like a ggplot figure that you can use in your reports or presentations. You can even do this programmatically, meaning you can write a script to automatically generate these refined figures without ever opening the interactive tool again. Now, let's talk about some real examples. The paper walks through three specific scenarios where ggsem shines. The first is for something called multi-group structural equation modeling, or SEM. This is used a lot in psychology to see if a model works the same way across different groups of people, like men and women, or different age groups. Usually comparing how parameters are different or the same between groups is done manually. With ggsem, You can visualize these differences directly and have all those visual choices saved. For example, in one group, you might highlight which parameters are allowed to change freely, and in another group, you might highlight which parameters are exactly the same. The second use case is about blending different ways of working. Many scientists do all their statistical work on scripts, which are basically sets of commands in R, but then to make pretty pictures, they might use interactive tools. ggsem bridges this gap. You can take a model you've already estimated using scripts, import it into ggsem, and then do that detailed visual tweaking. They show how you can even overlay two different models on the same diagram with different information displayed for each group, all while keeping track of every edit. This figure shows exactly that. On the left, you have a standard plot made with a script. Then in the middle, that same plot is loaded into ggsem where you can interact with it more deeply. Finally, on the right, you see an overlaid diagram where you can see both models at once, highlighting specific results like a significant difference in the residual variance. That highlighting is tied directly to the statistical test. The third use case is super powerful. It is about comparing models from completely different statistical approaches. Imagine you have data and you analyze it using regular statistics, like frequentism, then again using Bayesian statistics, and then again using network analysis. These different approaches often produce very different looking diagrams. ggsem lets you bring all these different types of diagrams into one place. In this example, they take models from 3 different packages: Lavan for frequentism, Blavine for Bayesianism, and QGraph for network analysis. They then use ggsem to make these very different diagrams look cohesive. They harmonize the colors so that if a certain variable is in a particular group in the network, it gets the same color in the SEM diagrams. They also align the layouts so you can visually see how the structures compare. This is huge for getting a complete picture of your data. This figure 4 illustrates this point perfectly. On top, you see the original separate diagrams. They look quite different, right? But after loading them into ggsem and doing some work, the bottom part shows a much more harmonized and comparable set of visuals. You can actually see how the different models relate to each other. So, what is the big takeaway from all of this? ggsem is a tool that brings clarity and reproducibility to scientific visualization. It moves beyond just making pretty pictures to creating visuals that are deeply connected to the underlying statistical models. Table 1 in the paper compares ggsem to other ways of making visualizations. What stands out is ggsem's ability to handle multiple models, its direct linking of visuals to statistical parameters, and its comprehensive metadata capture for reproducibility. While other tools are good at making single diagrams or require more manual work for comparisons, ggsem aims to centralize this comparative process. The core innovation here is that ggsem treats the statistical parameter, the actual number or relationship in your model, as the main thing you interact with. This is huge because it means that no matter how different your models are, whether they have different groups, different ways of framing the statistics, or even come from different statistical methods, the way you adjust their visuals stays consistent and meaningful. It helps you really dig into those subtle differences in your findings. The paper also talks about how ggsem differs from other graphical interfaces. Those are often great for building and estimating models in the first place, but ggsem focuses specifically on the visualization part after estimation. And it does it in a way that's reproducible and exportable to standard formats like ggplot. And how is it built? It's a local application, meaning it runs on your computer, built using something called Shiny. This is important because it means your data and your work never go out to the internet, unless you choose to share it. Everything is handled right there in your R session, keeping things private and secure. And again, all the work you do can be saved as metadata files and then used to regenerate your visualizations later, even outside of the interactive tool. Of course, no tool is perfect. The paper mentions that ggsem is mainly for creating static, publication-ready figures. It's not about creating interactive websites or live dashboards, though it integrates tightly with our workflows. Also, while it tries to be compatible with many different SEM packages, sometimes there are specialized features in those packages that might not have a direct equivalent in ggsem. This is a common challenge when trying to build bridges between different software tools. They also point out that, like any R visualization, full reproducibility depends on using the same versions of software and packages, something that can be managed with tools like Docker. Looking ahead, they plan to support even more modeling types, make the tool even easier to use, and study how people actually use these hybrid script guy workflows. They also mentioned considering background saving so you don't lose work if your R session crashes. What I find really exciting about this research is how it tackles that crucial but often overlooked step of making scientific visuals trustworthy and repeatable. For us as humans, We rely heavily on visuals to understand complex data. This tool helps ensure those visuals are not just pretty, but also accurate and transparent. It's a move towards more robust and verifiable scientific communication. It means that when scientists show us a diagram illustrating how something works, we can have more confidence that it's not just one person's interpretation, but a reproducible insight. The real-world implication is better science. When scientific findings are easier to reproduce and visualize accurately, it speeds up discovery. It allows other researchers to quickly build on that work, or to challenge it with more confidence. And it helps us all understand the complex world of psychology and other sciences a bit better. So, what do you think about ggsem? Does this sound like a tool that would have made your life easier if you've ever worked with scientific data or visualizations? Let me know in the comments below, and if you enjoyed this deep dive into making science more reproducible, make sure to give this video a thumbs up. Subscribe to RUBIN'S Mind Lab for more science breakdowns and hit that notification bell so you don't miss our next episode. Thanks for watching, and I'll see you next time.